Hello and welcome everyone to Science Fair 2020 and 21. This year's Science Fair is a bit different because of the pandemic, as we all know. However, the Science Fair is happening, and what can be better than that? My name is Mohammad Osman and I'm a student at A-levels in HURDCO International School. On 7th of October, these two wonderful women have been awarded the Nobel Prize in Chemistry by the Royal Swedish Academy of Sciences. They have been honored by this award because of their extensive research on CRISPR-Cas9. Ms. Amadou Carpenter and Ms. Jennifer A. Doudna. CRISPR-Cas9 has changed the concept of genetic technology. It is thought as one of the best research of the 21st century and is also considered as a new wave of science. So, from where this medical marvel came from? How does it work? And most importantly, what can it do? Surprisingly, CRISPR is a natural process that works in a bacterial immune system. It actually protects a bacteria from invading viruses. Naturally occurring CRISPR consists of two main components. The first one is a short, repeated DNA sequence called clustered regularly interspaced palindromic repeats, or simply CRISPR. And the second one is a Cas. A Cas is a CRISPR-associated protein that cuts molecular DNA like molecular scissors. So now the question comes, how CRISPR-Cas9 actually helps a bacteria to defend against an invading virus? To answer this, we need to see what happens when a virus actually invades a bacteria. So, when a virus actually invades a bacteria, Cas protein cuts a viral segment of the DNA of the virus and attaches it to the CRISPR region of the bacteria. As we can see that the cutout segment has been attached to the CRISPR region of the bacteria. From there, RNA starts to produce the RNA molecules. The RNA molecules produced attaches to a special protein called Cas9 to form a complex. This complex keeps on floating inside the bacteria and is in search of that virus. So, when the virus invades the bacteria for the second time, the complex immediately recognizes the virus and simply destroys it. So, a lot of bacteria surrounding us use this mechanism of CRISPR-Cas9 to protect themselves from invading viruses. However, in 2012, scientists discovered a way to hijack CRISPR-Cas9. And from that onwards, CRISPR has become the best genetic tool we have. But the question comes, how CRISPR works in a lab? To answer this, scientists use a Cas molecule and add a guide RNA and a templated DNA to it. The Cas molecule is then inserted inside a particular organism. The guide RNA guides the Cas molecule to the targeted gene, and the templated DNA commands the cellular protein to rebuild a new gene or to modify the gene. As we can see, a new DNA is formed, and over here the DNA is being modified. So, building a new gene or modifying the gene actually creates a lot of new treatments for many diseases. For example, we can create new treatments for sickle cell anemia, we can create new treatment for cystic fibrosis, and most importantly, we can create new treatments for cancer disease and can save life of a lot of people. The usage of CRISPR-Cas9 is endless. Researches are still going on, and we hope that we get all the benefits of CRISPR-Cas9 as soon as possible. So this was what I have to say about CRISPR-Cas9, and I conclude my speech here saying thanks to all my teachers who have arranged this beautiful program, and also thanking all the spectators of this video. Thank you very much.